coming into this molecular biology, rDNA technology, recombinant DNA technology, and lot of investigative technologies also coming in the molecular biology, right? Even vaccinations, uh, rDNA technology vaccinations like hepatitis B, hepatitis C, HPV vaccines, rabies vaccines. So a lot of vaccines were developing using this rDNA technology, right? Recombinant DNA technology. And yeah, this is one part of the biochemistry. Biochemistry means it's about the biomolecules, right? So what are the biomolecules here? Carbohydrates, proteins, lipids, and the fourth one is the nucleic acids, right? So the study related to nucleic acid comes under the molecular biology only. Okay. Okay. So if you know the DNA structure, then it will be easy for you to understand the purine and pyrimidine, because in the DNA and RNA, the purine and pyrimidines are existing. Okay. So if you know, or if you have some prior knowledge of the DNA, then you will understand a bit easily. Or if you have any doubt, just let me know. I will clarify. So today's topic is the purine and pyrimidine metabolism and disorders. So the class will be 2 hours, and in between we'll have a break for 10 minutes. Okay. Now let us see. So human body contains lot of cells, right? Human body contains lot of cells. So let's take a human cell, maybe a hepatocyte or liver cell, any cell. Let's take a cell. Cell contains the nucleus and also extra nuclear components like mitochondria, endoplasmic reticulum, Golgi apparatus, cilia, like that, right? So the nucleus. So the nucleus contains chromosomes. Okay. So you know about the chromosomes. So chromosome contains coiled chromatid. It contains the genetic information, right? So when you expand the chromosomes, then what happens? There is a double helix structure will be fine, right? So how this double helix structure? I mean, it's like a thread-like structure coiled together to form into one chromosome. So we have a lot of chromosomes, right? 23 pairs of the chromosomes, right? So out of 23, one is the allosome, remaining are the autosomes. I think you know about this one. Then this DNA structure is coiled, means around the histone protein, the DNA structure is coiled and it will compacted as a chromosome. So when we expand the chromosome, okay, the DNA structure looks like this. So this is called the double helix structure of the DNA. Okay, I think you know about this double helix structure. It looks like a ladder. Okay, so there are two strands. Okay, two strands moving opposite or like coiled oppositely, coiled in a opposite direction. So these two strands, put together with some bonds, right? So those are the double bonds and triple bonds. But basically, one strand contains the nitrogen bases. So what are the nitrogen bases here? Adenine, guanine, cytosine, and thymine. Okay, we'll see in the next slide. The central dogma of biology. So what do you mean by the central dogma of biology? Central dogma of biology means first DNA. What is DNA? It's a deoxyribonucleic acid. It carries the genetic information in the form of the genetic code. You know this one, right? So if DNA creates one more DNA, means if a cell multiply, then automatically the DNA also will get multiplied and it will be passed to the another cell. Okay, so DNA to DNA is called the replication. Okay, if one DNA is producing another DNA, that's called the replication. Then if the DNA is producing the RNA, then what is that RNA is called? DNA to RNA is called the transcription, right? Then from RNA to protein synthesis, it's called the translation. Okay, let me show one more slide. DNA to DNA is called the replication. DNA to RNA is called the transcription. Then RNA to protein is called the translation. How the proteins were created? Proteins will be developed or proteins will be synthesized in the body based on the genetic information present in the DNA, right? So DNA, it won't directly synthesis the protein, right? So there is a messenger RNA. So what the messenger RNA do? The messenger RNA carry the information present in the nucleus. Okay, outside, I mean, from the nucleus, it brings the message to the outside of the, I mean, nucleoplasm, that is the cytoplasm, right? From nucleus, it will bring the information into the cytoplasm. Then in the cytoplasm, so based on the code present on the mRNA, various types of protein will be created, right? So here, what is the central dogma? Central dogma means DNA to RNA, then RNA to the protein. So DNA to RNA is called the transcription. Then from RNA to formation of the protein is called the translation. Then what is the replication here? Replica means same, right? So DNA to DNA. The DNA can produce the same replica of its one more DNA, right? So when the cell is dividing, okay, or when there is a cell division happens, then it has to distribute the DNA. So it will give the same replica of the DNA to the other cell also. That's called the replication. Then what is the reverse transcription? Reverse transcription means, see, some viruses are there. Okay, they contain no any DNA. Then what happens? It contains only the RNA. Okay, so to produce the RNA, RNA cannot produce the RNA, right? So it converts the RNA to the DNA and then DNA to the RNA. Okay, so that's called a reverse transcription conversion, or the formation of DNA from RNA is called a reverse transcription. What is the transcription here? DNA to RNA is the transcription. Again, RNA to DNA is called a reverse transcription. An example of the reverse transcription enzyme or reverse transcriptase enzyme is present in the retrovirus, that is a HIV virus. HIV is a RNA virus. So that is the reason we call it as a reverse transcriptase, an enzyme is present, okay, because it forms the DNA from the RNA. Okay, that's the reason we call it a retrovirus. Retro, okay, retro means reverse, RNA to DNA. So this is a central dogma. Now, when you look at the DNA, so you know, right, the DNA is a double stranded structure. Okay, so two, phosphodiester bonds, we call these as the phosphodiester bonds, right? So when you expand it, okay, when you expand it, or if you make it, or if you drag it like a, ladder, it appears like this. It appears like this. So what are the, I mean, what's the content of this DNA here? See, uh, this part, this part, A, okay, A, and this is the sugar molecule, okay, maybe deoxy sugar or ribose sugar, and this is the phosphate. So this is the phosphate, this is a sugar molecule, and this is a nitrogenous base. So this nitrogenous base, sugar molecule, and the phosphate, these three will be considered as a nucleotide. What is it? Nucleotide. Okay, then if you consider only the adenine, that's the nitrogen base, and the sugar, then this is called the nucleoside, right? So nucleoside with phosphate is called the nucleotide. So what is this structure called? It is having multiple nucleotides, right? See, this is one nucleotide, and this is one nucleotide, this is one nucleotide, this is one nucleotide. So that is the reason we call it a nucleotide. It's a chain of the nucleotides, right? Then up, this is a double stranded one. So that's the reason opposite to this nucleotide, one more nucleotide is linked, right? One more nucleotide is linked. So what is this nucleotide? Always T is linked with the A with a double bond, means thymine is linked to the adenine and guanine is linked to the cytosine. So thymine, adenine, the hydrogen bond is double bond, and here the bond between the guanine and cytosine is triple bond. So as I told you, these are reversely coiled. Reverse means inversely, right? So one is coming from 3-5 direction, the other one is coming from the 5-3 direction. So oppositely they are coiled. So that is the reason here you can see the adenine, then it's a sugar, and this is the phosphate, right? So this is a nucleotide, and this is one nucleotide. Okay, so simply we can call it as it's a chain of the nucleotide or multi-nucleotide chain. Okay, now let us see here the same thing. What is the structure of the double-stranded DNA? So what is this? Here we have the deoxyribose phosphate backbone. Okay, so this is the phosphate, and this is the sugar, ribose sugar, right? Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So this is the backbone. Here also same, but here this is the 5-3 direction, and this is the 3 5 direction, opposite direction. See, it is coming from 5 3 direction, and it is also coming here from the opposite direction, oppositely coiled. So this is the backbone of one strand. This is the backbone of the other strand. So what's the backbone here? The phosphate and ribose sugar. Phosphate and ribose sugar. Okay. Then these two backbones are linked with a nitrogen base. What are the nitrogen base here? Adenine, guanine, thymine. Here, thymine, cytosine, then adenine means ATGC. The pairing is always ATGC, and the equal number of ATGCs will be there. Okay. When there are 10 ATs are there, then the same number of GCs will be there. Okay. Then these are linked with the double bonds. Then these are linked with the triple bonds. Okay. So these are the hydrogen bonds. So this is the link between the two strands, right? So two strands are anti-parallel in opposite directions. Then hydrogen bonds between bases of opposite strand, AT and CG. Then denaturation. So when can you find the single strand? See, this is a double stranded, right? This is a double stranded one. It's a coiled actually. So this is a completely coiled one. Okay. When you see this, it's completely coiled. So what we do in the laboratories, we use some denaturing agents so that after adding the denaturing agents, what happens? The nucleotides are the phosphodiester strands. Okay, these are the phosphodiester strands, right? So these strands will get separated, okay, based on the chemical compounds we use. So breakdown of hydrogen bonds between two strands leading to the formation of two separate single strands, right? So what the denaturation do? It will break the two strands. Okay, where it make the cutting, it cuts the base pair connection, hydrogen bonds, so that this will become a separate strand and this will also become a separate strand. So what are the methods of denaturation? Either you can use the heating techniques, or if there is any change in the pH, then automatically these two will get denatured and will separated into two separate strands. Okay. Then already we discussed this one. What are the components here? It contains a sugar that may be a ribose sugar or that may be a deoxyribose sugar. So this one, okay. Here you can see, right, pentagon. So what is this? This is either ribose sugar or deoxyribose sugar. So what's the difference between the, if it is an RNA, RNA contains a ribose sugar. If it's a DNA, DNA contains the deoxyribose sugar. Then it contains a phosphate group. It also contains a nitrogen base. Nitrogen base may be a purine or pyrimidine. So what is a purine? What is a pyrimidine? What is a purine pyrimidine? So we'll discuss now. You understood, right? It contains a backbone structure. What is the backbone? It's made up of phosphate and sugar. This is the backbone. And to the sugar, nitrogen bases were attached. So these nitrogen bases we classify into two types. One is purine, the other one is a pyrimidine. Okay. Then what is the difference between the nucleotides and nucleosides? Already this I explained. Nucleotides means this structure is called the nucleotide. Means all together we call it a nucleotide. Sugar, nitrogen base, and the phosphate. These three we call as a nucleotide. Then what is a nucleoside? Nucleoside means it contains only the sugar. Okay, see, nucleosides means it contains the sugar and the nitrogen bases. That's it. So we are not including the phosphate in the nucleoside. Okay, so when it is a phosphorylated, then that is a nucleotide. When there is no phosphorus or when there is no phosphate, then nucleoside. Clear? Now see the definitions once again. Nucleotides means nitrogen base plus sugar plus phosphate group is called the nucleotides. Nucleoside means it contains no phosphate group. It's just nitrogen base and sugar. Then how many types of nitrogen bases are there? We have two types of nitrogen bases, purine or pyrimidine. Two types. Then how many sugars are there? Ribose sugar and deoxy sugar. What is a purine? Purines are adenine, guanine. Pyrimidines are thymine, cytosine, uracil. There are three pyrimidines: thymine, cytosine, uracil. Then purines are two: adenine, guanine. Okay. So, but in DNA, we have only thymine and cytosine. Okay. When you take the RNA, so RNA contains no cytosine, it contains a uracil in the place of cytosine. Got it? So always in the double-stranded DNA, it contains the adenine, guanine, cytosine, thymine only, not uracil. When you take the RNA in the place of cytosine, RNA contains the uracil. Okay, if it is double-stranded, even though it is single-stranded, it may contain the uracil in the place of the cytosine. right? So these are the terms. Now coming to the nucleotide again, just for a better understanding. Okay, again I'm explaining the same thing. What's the nucleotide here? Phosphate, nucleoside, and nitrogen base. So these three are considered as a nucleotide. Okay, and how the sugar is linked with the nitrogen base? How the sugar is linked with the nitrogen base? There is a N-glycosidic bond. Okay, so this nitrogen base is linked to the nucleoside, okay, with this N-glycosidic bond. Okay, N-glycosidic bond. Clear? So, and one more thing here, there are, this is a phosphate, right? This is a phosphate group. But sometimes this nucleoside, okay, this nucleoside means, what is the nucleoside here? The base and nucleoside, right? The nitrogen base and the sugar, these two are considered as nucleoside, right? So sometimes the nucleoside is linked with only one phosphate. Okay, let us see the next slide. What are the purines? Adenine, guanine. What are the pyrimidines? Cytosine, thymine, uracil. DNA contains adenine, guanine, cytosine, thymine. Okay, RNA contains in the place of uh, in the place of thymine, uracil, right? Cytosine, uracil. Sorry. Uh, yes, yes. Okay, in the place of thymine, it contains uracil. Okay, not the uh, thymine, cytosine, uracil, right? Yes, in the place of thymine, it contains the uracil. Okay, not in the place of cytosine. Okay, then yeah. Now coming to the phosphate. Okay, see, this is a nitrogen base. This is a nitrogen base. This is a sugar. Okay, this is a sugar. So this nucleoside, when it is linked with one phosphate, then we call it as a adenosine monophosphate. Only one phosphate. If it is linked with the two phosphates, then adenosine diphosphate. Two phosphates. So when it is linked with the three phosphates, we call it the adenosine triphosphate. Okay, or triphosphate. So what do you mean by adenosine triphosphate? What do you mean by ATP? ATP means it contains the three phosphates along with adenosine. What do you mean by adenosine? What do you mean by adenosine? It is a nucleoside which contains a nitrogen base as well as a sugar. Okay, so what is this? This is the nucleotide. Clear? Then coming to the ribose sugar. So what is the difference between the ribose sugar and the deoxyribose sugar? So where the ribose sugar is present, it is present in the RNA. Then deoxyribose sugar is present in the DNA. So what's the difference between the sugars of RNA and RNA and DNA? In RNA, there is a ribose sugar. Okay, but in DNA, so there's a lack of this oxy. That is the reason deoxy means here there is no oxygen. Right here, OH group is there. Here there is no OH group. That's the reason we call this as a deoxyribose. So where this deoxyribose is present in the DNA, this is present in the RNA. Okay. Then bases. Okay. So what are the bases? Already we discussed there are two types of bases, purines and pyrimidines, which are present in the nucleic acid. Purines are double ringed structures, pyrimidines are single ringed structures. And again, this is very important. So how to differentiate the pyrimidines and purines? So there is one phosphate group, okay, just now we discussed there is a single phosphate, double phosphate, triple phosphate also like this. See, it's a nucleoside. When it's linked with one phosphate, there's a nucleotide or AMP. I mean, AMP, adenosine monophosphate, right? So when it's linked with the two phosphates, then nucleoside diphosphate. Third one, nucleoside triphosphate, Then coming to the purine and pyrimidines, always the purines are double ringed structure. See, it's a double ring, and pyrimidines are the single ring. Okay, single ring. Okay, so how to remember this double ring and single ring here? Pyrimidine, single rings, purines, double ring. So actually, to remember, there is one mnemonic. I'll show you how to remember that mnemonic. Yeah. So purine means pure as two gold rings. Pure as two gold rings. Just remember this one. Pure means purine. Purine. Pure means purine. As two gold rings, right? As means adenine. G means gold ring, right? G means guanine. So the purines are adenine and guanine. Okay. Simply remember this one. Purines are pure as two gold rings. Pure as two gold rings means it's a purine. Okay, as means adenine, G means guanine. So purines are adenine, guanine, and it is a two gold rings, right? So that is a double structure. Okay, it is having double structure or double carbons. Okay, double rings. Okay, that's the reason we are using this two gold rings. Okay, so this is how you have to remember the purine. Then what's the pyrimidine? Anyway, that's a, I mean, after purine, there is only one pyrimidine. That is a single ring structure only. Okay. So pyrimidine, you can easily remember like this. Then, and one more thing, just now we discussed, thymine is present in the DNA, uracil is present in the RNA. Okay. So the DNA may not, DNA, I mean, it contains no uracil, right? So in DNA, it contains the thymine and uracil, and you know the base pairs also ATGC. Always adenine linked with the thymine and guanine links with the cytosine. Okay, they're linked with the hydrogen bonds. Okay. Now, if adenine, adenine monophosphate. So what is this one? Adenosine, adenosine. See, you know this one, right? What is this? This is a nucleotide. Nucleotide contains a nitrogen base and a sugar and one phosphate, right? So based on the type of the nitrogen base linked to the sugar, the name of the nucleoside and the name of the nucleotide will change. Okay. See, for example, here in the place of base, I mean base means nitrogen base, adenine. We have adenine, guanine, cytosine, thymine, uracil, right? So if the nitrogen base is adenine here, then what we call it as a adenine, okay, uh, see, adenosine, right? If it is a single one nitrogen base, then that is a adenine, right? But when it is linked with the sugar molecules and the phosphate, or let us see the nucleosides only. See, uh, adenosine, adenosine. See, adenosine means what is adenosine here? Adenosine monophosphate. Adenosine plus one phosphate. What is this adenosine? Adenosine is a what is this adenosine? Adenosine is a nitrogen base with sugar. Okay. Then if you are adding one phosphate, then that will become adenosine monophosphate, AMP. If the nitrogen base is guanosine, okay, or guanine plus sugar, guanosine, then guanosine monophosphate. If it is cytidine, then cytidine monophosphate. If it is uridine, what do you mean by cytidine? Cytidine means cytosine. You are adding the cytosine with the sugar, then that will become the cytidine. If you are adding the thymine with the sugar, then that will become the yeah uh, yeah. Here you can see because these are in the RNA, these are in the DNA, right? So what do you mean by uridine? What do you mean by uridine? Uracil plus sugar. Okay, that is the uridine plus phosphate you are adding. So uridine monophosphate. Clear? Then in the DNA, adenosine plus phosphate. So DAMP, deoxyadenosine monophosphate. Here it is a deoxyribonucleotides, right? So that is the reason we are using the D. Okay, deoxyadenosine monophosphate. Then deoxyguanosine monophosphate. Then deoxycytidine monophosphate. Deoxythymidine monophosphate. Got it? So these are the ribonucleotides and these are the deoxyribonucleotides. See, for example, in the place of AMP, okay, if you are using ADMP, what do you mean by ADP? Adenosine diphosphate. Okay, means here it's not linked with one phosphate. If it is linked to two phosphates, then we call it the adenosine diphosphate. Okay, so what are the functions here? Nucleotides are precursors of nucleic acids, right? See, to form the nucleic acid, then definitely we should have the nucleotides, right? With the nucleotides only, the nucleic acid may form. What is a nucleic acid? It's nothing but DNA, RNA, right? So nucleotides are the precursors of the DNA and RNA. The nucleic acid are concerned with storage and transfer of the genetic information. Nucleosides are used as antiviral and anti-cancer agent in the medicine. Nucleosides. Okay, I will explain how these are used as the antiviral and anti-cancer agent in the medicine. Okay, then coming to the some more functions, the universal currency of energy, namely ATP, is a nucleotide derivative only. ATP, energy. Okay, so it's a universal currency of energy in the body, right? So for every metabolism, we need the ATP. So ATP is the derivative of the nucleotide only. Okay, so nucleotides are precursors of important coenzymes like NAD. These are the precursors. Nucleotides are precursors of the because you know, right? For the formation of NAD, FAD, we need the nucleotides, right? Metabolic regulators such as CAMP and CGMP also, we need the nucleotides. So these are the functions of the nucleotides, right? Then again coming to the clear and detailed structure of the nucleotide, because today we are learning the metabolism of nucleotides. We should know the complete structure. That's the reason I am stressing these many times. Okay, the structure of the nucleotide. So it is a sugar, then this is a phosphate. If it is only one phosphate, then it's a AMP, adenosine monophosphate only. Then this is the base. Okay, what is this base? It's a nitrogen base. Then how it is linked with the sugar? Glycosidic bond, N-glycosidic bond, right? N-glycosidic bond. Okay, then here OH. OH means it is having the oxygen, so ribonucleic acid. When it is oxygen is removed, then it's called the deoxy. Okay, that's the reason deoxyribonucleic acid. Okay, this is the difference. Okay, so it's a structure of a nucleotide. Then what is the link between this sugar and the phosphate? Phosphate ester bond. Phosphate ester bond. Okay, phosphate ester bond. What is the link between the base and sugar? That is glycosidic. N-glycosidic bond. This is a phosphoester bond. Okay. Then coming to the pyrimidine, you see the complete structure here. 1, 2, 3, 4, 5, 6. Okay. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Because purine, it is a double ringed. Just now we discussed. Purine means pure as two gold rings. Pure as two gold rings, right? So it is having two rings. This is having only one ring. Okay, purine. Then actually adenine, right? So this is a molecular term we should use. Okay, I mean simply we are using the adenine, right? But you can use this word 6-aminopurine. What is adenine called? 6-aminopurine because here you can see in the sixth place it is having NH2, right? That is the reason we call this the 6-aminopurine. Here in the sixth place it's having the amino group, right? 6-aminopurine. Then guanine is also called as the 2-amino-6-oxypurine. Why? Because in the second place it is having an amino group, and at sixth place it is having a oxygen group, right? Oxy group. That's the reason we call the guanine as 2-amino-6-oxypurine. Okay, these two are the purines only. Okay. Then coming to the thymine. So thymine we call it the 2,4-deoxy because in the second place it is having oxy, and the fourth place also it is having oxy. So 2,4-deoxy, right? Then 5-methyl pyridine. So this is a 5. I mean fifth place, fifth carbon, there is a methyl group. So that is the reason 5-methyl pyridine. Okay. Then what is this cytosine? Cytosine means 2-oxy. Second place oxy, then 4-amino. In the fourth place there is an amino pyridine. Then what is this uracil? In the uracil, in the second place as well as the fourth place, both are having the deoxypyridine. Okay, so these are the molecular names or molecular formulas, right? Molecular structures. So today's session we are talking about the biosynthesis of purine and the pyrimidine also, both. Not only biosynthesis, even the degradation also we are talking. Okay, purine biosynthesis, then pyrimidine biosynthesis. Then in the purine biosynthesis, we are talking about the de novo synthesis. And in the purine biosynthesis, we are talking about the salvage pathways. Both are having the de novo and salvage, but in the purine biosynthesis, de novo is the best one, and we are also talking about the salvage also. Okay, but in the pyrimidine biosynthesis, basically salvage pathway is important. Okay, now what is the difference between this de novo and salvage? Okay, this is a very important one. What do you mean by de novo and salvage? De novo means it's a new car. New car. New car means it's a new one. Okay, you are creating a new one. De novo means you are creating, it's a Latin word, right? So you're creating a new one. Salvage means you are creating from the old one. Okay, means see, we are taking the nucleotides. Okay, or when you eat some meat, meat contains the nucleotides, right? After breakdown of the nucleotides, the nitrogen bases will come. Okay, then we are using those nitrogen bases again to build a new nucleotide or new DNA or new RNA. Okay, means utilizing the nitrogen bases directly, okay, to build the new DNA or new RNA. So that is salvage. What is a de novo? De novo means we are creating the purine and pyrimidine on our own, adding the nitrogen group, using the glutamate, or using some glycine, okay, or using some aspartate, or using some formyl, or using some methyl group, okay, or using some carbon dioxide. Means we are using different compounds, okay, uh, or different compounds are contributing to the creation of new purine or pyrimidine. That is called the de novo. So there are two types. One is the de novo. De novo means we are using different compounds, right? What are the compounds? We have different compounds, like whether you use a formate, glycine, you know, because a lot of things will be contributed to the creation of one purine and one pyrimidine also. Got it? So that's a new creation of the pyrimidine or purine. Then salvage means already existing. Means after breakdown of the, the purine or the breakdown of the nucleic acid, we are reutilizing that one. Okay, that's called the salvage. Clear? Now, coming to the difference between the purine and pyrimidine synthesis. So, purines means purines in the, I mean, we are talking about the synthesis, right? The purines and pyrimidine synthesis. So, when you see the difference between the purine synthesis and pyrimidine synthesis, so you know this one, right? This is a phosphate, this is a sugar, and this is a nitrogen base. So, we are talking about the nitrogen base, right? We are talking about the synthesis of the purine, right? We are talking about the synthesis of the purine only, right? So here already the carbon is there. Already, the sugar is there, right? It is already having the phosphate. So when it is synthesizing the purines, when it is synthesizing purines, what happens? Already the carbon is, already there is a sugar. See, synthesis starts with PRPP. Purine ring is built step by step with the C1 of the PRPP as a primer. Got it? Means it will build step by step on the sugar. Okay, on the sugar. It will build step by step. But here pyrimidine means initially it will build the ring and then it will add it to the ribose sugar. Clear? What is the difference between the purine synthesis and pyrimidine synthesis means on a sugar molecule, the step by step, the purine will be built. But coming to the pyrimidine, initially the ring will be built and then it will be added to the ribose. Okay. So this is the difference. Now, yeah, this already we discussed. Then coming to the biosynthesis of purine nucleotides. So purine nucleotides consist of purine base. Again, the same content. Okay, just a repetition. Purine, what is the nucleotide? It contains the sugar. Okay, that's a pentose sugar, maybe a ribose or deoxyribose, and one molecule or more than one molecule. So the phosphate maybe monophosphate, diphosphate, or triphosphate, and a purine base. What are the purines here? Already we discussed about the purines, right? Gold rings, guanine, adenine, right? So adenine, guanine is the major purine bases, but along with this major purine bases, we have some other purines also. Those are called the minor purine bases. What are the minor purine bases? Hypoxanthine and xanthine. Okay, adenine, guanine are the major purine bases, but we have minor purine bases also, hypoxanthine and xanthine. So these purine bases combine with pentose sugar and phosphates to form the purine nucleotide. This we know. Okay. Then coming to the purine nucleotides, how many types of purine nucleotides are there? Already we discussed. If it is a adenine, if it is linked with one phosphate, monophosphate, so how many adenines are there? AMP, ADP, ATP. How many guanines are there? GMP, GDP, GTP. Okay, monophosphate. So adenine three adenines are there: ADP, AMP, ATP. Then coming to the guanines, GMP, GDP, GTP. Okay. Then we have minor purine bases also, right? Hypoxanthine. We call this hypoxanthine as the IMP. IMP means inosine monophosphate. Then XMP also. XMP means xanthine monophosphate. Okay. Hypoxanthine, xanthine. Okay. If it is a hypoxanthine, then IMP. If it is a xanthine, then xanthine monophosphate. Clear? So totally how many purine nucleotides we have? 3 plus 3, 6, then 7, 8. Including the minor purine bases, totally we have 8. Clear? Okay. So now we have to discuss about the biosynthesis, how these are synthesized. Okay. So where these, as I told you, we have two methods. One is de novo, the other one is the salvage, right? First one is the de novo. So we're discussing the de novo. De novo synthesis means we are synthesizing newly. Okay. So what is the site of synthesis of this nucleotides in cytosol of liver, small intestine and thymus? Okay. Small intestine, thymus and liver cells. Liver cells, right? Cytosol. What is the cytosol? It's not the, it is not within the nucleus. Okay. It's not within the nucleolus, right? It is in the cytoplasm. Cytosol is nothing but the cytoplasm outside of the nucleus. Okay. Then coming to the characteristics, purines are synthesized using 5-phosphorypose as a starting material step by step. So where it starts from? 5-phosphorypose. I will show you. PRPP, 5-phosphoribosyl 1-pyrophosphate is the active donor of R5P. AMP and GMP are synthesized further at the base of IMP. Okay, let us see this one. See, this is one purine. Purine. Why purine? Two rings. Two rings, right? Two rings. That's the reason it's a purine. So maybe it is a adenosine, maybe guanosine. But basically, to create one purine, what we need? We need a lot of compounds. Okay. One is aspartate amine, which is an amino acid. We need a formate also. We need a glycine. We need bicarbonate. We need formate. We need ammonia. We need nitrogen, right? Nitrogen means it's nothing but amino acid, right? So all these are contributed to create one purine nucleotide. Got it? So we need all these things. So where all these things will come from? From our food source only, right? From our food source only. So we are not utilizing the existing purine as it is, just like the salvage, right? Here we are creating. Here we are creating using multiple compounds or different compounds. Clear? So what is the what are the different components present in the purine means? These are the different compounds. Formate, aspartate, glutamine, okay, formate, glycine, bicarbonate, like that. So we need different compounds to create a purine. Okay. Now there are three steps to create the purine nucleotides. There are three steps. One is IMP formation. What is IMP formation? Already I told you, inosine monophosphate. What is the inosine monophosphate? It is nothing but xanthine. See, inosine monophosphate means hypoxanthine. First, it creates the hypoxanthine. Okay, first, what it do? It creates the hypoxanthine. Now, see, IMP formation. First, it creates the inosine monophosphate. It's the first nucleotide synthesized. Then from this IMP to AMP and GMP. Okay. Initially, it creates an IMP. Okay. Then from IMP, it creates the AMP and GMP. What's an AMP? Monophosphate, adenosine, and guanine. So AMP is then converted to AMP and GMP. And later, AMP will be ligated. Okay. AMP will be kinase, kinase, right? Not ligated, kinase. So this AMP will be kinesed and converted to the ADP and ATP later. And GMP will be kinase to the GDP and then GTP, like that. Clear? So these are the three steps. Initially, hypoxanthine will form. Hypoxanthine monophosphate. There is nothing but inosine monophosphate. And later, AMP and GMP will form. So these are the three steps. Clear? Now, coming to the basic pathway. Starts from ribose 5-phosphate. Ribose 5-phosphate means sugar and phosphate, right? Sugar and phosphate, because we are creating a nitrogen base, that is the adenine or guanine. It needs total 11 steps overall. It needs total 11 steps. Okay. Where it happens occurs primarily in the liver or intestine. Okay. Now, this is the complete pathway. Okay. It's a complete pathway. Then you can see there are 11 steps are there. 11 steps are there. Okay. So I'll be explaining step by step. So first, this is the sugar. It's a pentose sugar, maybe ribose. Okay. Yeah, here we have OH means is ribose, right? Ribose 5-phosphate. Ribose 5-phosphate, and this is the, ribose 5-phosphate means at the fifth position it is linked with the phosphate. So that is the reason it's called the ribose 5-phosphate. Okay. Then after adding the ATP, it will be converted to the, 1 AMP will be released. Okay, 1 AMP will be released. Means 2 phosphates. Here 3 phosphates, right? So one is liberated, 2 phosphates were added here in this place. Okay, so this structure is called the 5-phosphoryl 1 pyrophosphate. So just remember this as the PRPP. 5-phosphoryl 1 pyrophosphate, PRPP. Okay, 5-phosphoribosyl 5-phosphoribosyl 1, or we also call it alpha. In the place of 1, we call it alpha also. Alpha pyrophosphate, PRPP. So initially what happens? The pentose sugar with phosphate with the ATP, this structure will form. What is this structure called? The 5-phosphoribosyl alpha pyrophosphate or 1 pyrophosphate. Okay. Then from here, the next scene will start. Okay. Yeah. Can you see here this side, right side on the top? Okay. Already I told you these compounds are required to create a purine. What are the compounds? I told you formate, aspartate, glycine, formate, then glutaminamide, then this is the bicarbonate, like multiple things we need. But in the first step, what happens? See, in the first step, what is happening here? It's the PRPP. PRPP means it's a P, right? See this? Here, this is a P and this is a P. What is this called? This is called the PRPP, 5-phosphoribosyl alpha pyrophosphate, right? Then here, the glutamine is getting added. Glutamine. Here you can see we need the glutamine also, right? Glutamine. After releasing the PPA, then it will be converted to glutamate that will be liberated. So what this step is doing here? This step is adding N, right? N in which place? See, committed step of the pathway. An amino group donated by glutamine is attached at C1 of the PRPP. So this is a 1, right? C1. This is a carbon 1. See, I don't know whether you remember it or not. That is the reason I explained you with the structure. Okay. See, where is the carbon 1 here? It's a carbon 2, right? It's a carbon 1, 3, 4, 5, 6, 7, 8, 9. So it's a carbon 1 place. Okay. Okay. So, yeah. So is attached at C1 of the PRPP resulting in 5-phosphoribosyl amine. Okay. So this PRPP, after the addition of this glutamine, okay, after addition of this glutamine. So yeah, glutamine is attached at C1. Okay, C1 of PRPP. Okay, resulting in 5-phosphoribosyl ribosylamine. Here there is one enzyme. Okay, because without enzyme, no step will move further, right? For every step, there is a role of enzyme, right? So for conversion of each step, we need the enzymes. So what is the enzyme here? Glutamine PRPP amido transferase. Glutamine PRPP amido transferase. So what is doing? It is transferring the glutamine, which is an amino acid, right, to the PRPP. What is this enzyme called? The glutamine PRPP amido transferase. Uh, I think somebody want to talk to me. Yeah, if you have any issue, just ping me here. Okay, I will respond. So attendance is 41. It's good. Good. Okay. I hope you are listening. Okay. Yeah. So here, carbon one place, right? C1 position. Which one is glutamine? Okay. Glutamine is linked to the carbon one place. Now, coming to the second step. In the second step, what is happening? Addition of three atoms from glycine. Okay. See, this is a sugar, right? This is a sugar. This is a phosphate. It has to create one. See, it has to create this complete one, right? It's a complete one. So, what is this place? N1. I mean, it's a carbon one, right? So that is the reason it is adding here. It is considered as the carbon one place, C1 place, C1 place. From here only one by one, it will add and it will makes two rings here. This one, this one. Okay, it has to create two rings. So initially it created or it added an amino group here. Okay, what is it? Glutamine here in the carbon C1 place. Then later, yeah, then later in the second step, what is happening? Addition of three atoms from glycine. Okay, glycine. What are the three atoms here? Can you see? NCC means 457 places. 457 places were added. Okay, initially this got added N. So to this N, CCC means 4, then 5, then 7. Okay, 457 were added. Okay, 457 were added. Okay, what is this enzyme here? GAR synthesis. What is GAR? Glycosinamide ribonucleotide enzyme or synthesis. Okay, now this one formed and this glycine also formed. Okay, now I think this one, right? N means this one. I think that is C1 or this N, right? N means I think C9. Okay, leave it. I will explain that later. You understood, right? So acquisition of purine at C4, C5, and N7. Okay. So in the initial stage, one molecule added. Okay. Then later, three were added. Three atoms, sorry, not molecule. Three atoms were added. NCC, fourth, fifth, and seventh. Then in the step three, the added glycine amino group is then formulated by N10 formyl tetrahydrofolate. So here in this third step, acquisition of purine atom at C here. Okay, with the addition of this formyl tetrafolate, see here the format is added here in the eighth place, means the carbon, okay, accumulated here. See, initially in the first stage, this got added. Second stage, this got added. Okay, now in the third stage, this got added. It has to create two rings. Okay, then coming to the fourth stage. In the fourth stage, what is happening? Accession of purine atom at 3 here, purine. Okay, what is the atom N3 here? That is again glutamine. Okay, glutamine in the N3 place. Okay, here. This structure, now again, here it added one more glutamine. Okay, later in the next step, closing of the imidazole ring. So this is one ring, right? So this is one ring, then it is closing. So how it is closing? Again with the help of the ATP only and with an enzyme. Later in the sixth step, it got modified or mutazed. Actually, in prokaryotes, like bacteria and fungi, right? Eukaryotes and prokaryotes, right? In eukaryotes, directly this fifth step, it goes to the it goes to the seventh step. Okay, seventh step. Uh, no, eighth step, right? So there won't be any six and seven steps. So directly from this fifth step means after closing this imidazole ring, so it will be converted. It will be, I mean, it's modified, right? It will change its shape. Okay, change its shape like this. But if it is in prokaryotes like bacteria and fungi, so some extra steps also will happen. Okay, so it will get modified. Simple. This is an imidazole ring. Okay, this is an imidazole ring. Okay, so it got closed. So after closing, the transformation may happen. Means the change of the carbons, I mean, from here to here. So in higher eukaryotes, including humans, 5 air product of step 5 is carboxylated directly to in one step instead of two. Okay, I mean instead of 6A and 6 and 6A, it goes to the 7. So what is happening here? Simple. Carboxyaminoimidazole ribonucleotide. So this is the structure formed. Okay, and I mean this is a mutate structure. Okay, so this is a structure. It got converted to this type of structure. Then later, okay, and later in this stage, what is happening? Acquisition of C6 and N1. Okay, so C6 means here you can see this one bicarbonate and N1 also. These two got added. These two got added here. Okay. Then this will be deleted. Okay, the elimination of fumarate. So this is called the fumarate. Actually, this is called the fumarate. So this fumarate will be deleted in the next step. This fumarate will be deleted. Okay. Then after elimination of this fumarate, then this type of structure will form. This type of structure will form, and later, I mean, anyway, it is linked to the sugar molecule, right? See, this structure got formed. Okay. So in the step 10, acquisition of carbon 2. Here we need the carbon 2 also, right? Here there is no carbon 2. There is no carbon 2. Then the carbon 2 got added. Then in the 11th step, ring closer to form the IMP. Okay, then this one after adding the carbon here in the second place, then ring called ring closed. Okay, so this is the two ring structure. Okay, which is linked with the phosphodized, I mean, N-glycosidic bond like this. So what is this? This is the inosinate. Inosinate means hypoxanthine, right? Hypoxanthine monophosphate. So finally, this structure got created. Clear? Slowly, one by one got added. Slowly, one by one got added, and finally, it created a two-ringed structure. That is a purine. So what is this purine? Initial purine formation is the inosinate. That is the IMP. Okay. So it happened in 11 steps. So if you don't remember all these, just simply write the names. So here I give the names. Initially, D-ribose, D-ribose 5-phosphate, right? D-ribose 5-phosphate. Then later it is converted to 5-phosphoribose 1-pyrophosphate. That is the PRPP. Okay. Then later glutamine got added. Then 5-phosphoribosylamine. Okay. This is called the PRA. And then ATP. Later it converted to glycosinamide ribosyl. Okay, that is formal glycosinate ribosyl. Later, again it converted. Again it is taking glutamine. Okay, ring closure in this stage. Later it's converted to biotin after adding the CO2, and again aspartate got added. Okay, where the imidazole ring closed. Then later fumarate. We release the fumarate. After releasing the fumarate, then finally after adding the carbon in the second carbon, then ring closure, it creates the inosinic acid. Okay, IMP. Then from this inosinic acid, two two nitrogen bases. One is the adenosyl, then xanthine monophosphate. Okay, so from this xanthine monophosphate, the guanosine monophosphate formed, and then adenosyl succinic acid, adenosine monophosphate. Okay, already we discussed, right? Initially, the inosinic acid will form. Then from the inosinic acid, xanthine monophosphate, and from the xanthine monophosphate, guanosine monophosphate. And from inosinic acid, again, it creates the adenosine monophosphate. This is the second step. Okay. So what in the second step, I mean, can you see this? I mean, the molecular structures. Initially, IMP formed inosine monophosphate, right? Means ribose sugar and two rings, inosine monophosphate. Then it converted to the adenyl succinate synthase. I mean, using this enzyme, it converted to the AMPS, and from the AMPS, adenyl succinate to AMP. Okay, or IMP is converted to the AMP, adenosine monophosphate, and again this inosine will be converted to xanthine monophosphate, and this xanthine monophosphate is converted to the GMP. Okay, the same thing. See, inosinic acid converted to xanthine monophosphate, guanosine monophosphate. Then this is converted to adenosine monophosphate. So XMP, GMP, AMP created four. Okay, one, two, three. Then inosinic acid nothing but the hypoxanthine. Four, right? So one, two, three, four. So four purines got created. Okay, then if it is linked with phosphates, then again they will be converted, right? Like this. The same thing, inosine monophosphate will be converted to adenosine monophosphate in this case, then xanthine monophosphate, and then guanosine monophosphate. Okay, if you don't remember these rings and structure, just simply write the notes. But for medical students, I think this is enough. This is enough. Okay, so no need to write. I mean, write all these rings. Okay, just mention the names. That is more than enough. Write the enzyme name as well as the compounds added in each step. Okay. Yeah. So yeah, AMP. Then what happens? AMP after adding one more. After taking one more phosphate, it's converted to the ADP. Then GMP will be converted to the GDP, and again after taking one more phosphate, it will convert to GTP. Okay. So what is our goal? We have to create all the purines. What are all the purines we learnt here? So this is a list of the purines. Purine means, yeah, these are the AMP, ADP, ATP. Okay, GMP, GDP, GTP, IMP. So first, what is happening? Initially, IMP got created. With that 11 steps, IMP got created. Then from that IMP, GMP, I mean XMP got created. Then from the XMP, GMP got created, and from IMP, AMP got created. Okay, so 1, 2, 3, 4, and later with the phosphate, AMP converted to the ADP and then ATP. Okay, that is nothing but kinase. Okay, we call this a kinase, NMP kinase. Then GMP will be converted to the GDP and GDP. Okay. So initially we need 11 steps to convert the, I mean, to create the IMP. Okay. Yeah. Then later, so this is a nucleotide, right? So this is a base pair. It's a nucleotide. What is this? This is a ribose. Okay. Then this ribose will be converted to the deoxyribose also. How it is converted to the deoxyribose? Using the ribonucleotide reductase enzyme or thioredoxin reductase. Okay, it uses the ribonucleotide reductase and thioredoxin reductase. Later, it will be losing this oxygen and it will be converted to the deoxyribose. Okay, deoxyribose nucleotide. This is the nucleotide. It's a deoxyribonucleotide. Okay. So what is this deoxyribonucleotide? It's present in the DNA, right? See. This one. This one. Okay. Here AMP is converted to the DAMP. How it is converted? How it is converted? How it is converted? Just now I told you, right? Ribonucleotide reductase and thioredoxin reductase. Okay. How many nucleotides totally? Because just now we learned de novo how to create the adeноnucleotides we are talking about, right? Nucleotides. First, initially, it creates the nitrogen bases and then it adds to the, I mean, from the ribose sugar only, it will add one by one compound and finally it will be creating a complete nucleotide, right? Then those nucleotides will be converted to the deoxyribonucleotides also with the process called the reductase. So here how it is reducting means it is removing the oxygen, reductase, right? Reduction. Okay, using the enzyme called the reductase. So what are the reductase enzymes we are using here to convert the RNA to DNA? Ribonucleotide reductase, then redoxyreductase. Yeah, here you can see ribonucleotide reductase and thioredoxin reductase. Clear? So this is about the synthesis of the purines. Purine means adenine, guanine, and other AMPs, DMPs, or uh, TPs. Okay. So then regulation. So how it is regulated? What do you mean by regulation? Regulation, I think you know, right? Regulation is very important for homeostasis, right? The pH should be regulated, the water balance should be regulated in our body, everything should be regulated. All the enzyme activities also regulated, even the growth also regulated. Okay, the secretion of the enzymes is regulated, the secretion of hormones is regulated. So if there is no regulation, then irregular development will be there. What does it mean by irregular? Cancer cell, right? There is no stop of the cell. Then it grows continuously. So there should be some feedback mechanism should be there. Feedback mechanism means, see, you are hungry. When somebody is feeding you continuously, then if you don't say that, I mean, when you don't say that I am not hungry or I am full, then till that time they will be feeding you, right? So till what time they will feed you? Till you express, right? I mean, if you say that I am okay, I am full, then they will stop, right? So feedback mechanism should come, right? So if you are not saying I am full, then they will be continuously feeding you only. There won't be any regulation, right? It will be continuous. So in the same way, here also, the regulation should be there. I mean, in the purine nucleotide, biosynthesis will be there, but something is regulated. So I mean, when you see the total, we have 11 steps, right? See, first one is ribose 5-phosphate. First one is ribose phosphate, means it contains a ribose sugar and a phosphate. Then later it's converted to the PRPP. I told you, right? Phosphoribosyl alpha pyrophosphate or 1-pyrophosphate, right? So here how it is regulated? The entire ribose 5-phosphate will not be converted to the PRPP. Why? Because once we have sufficient ATP, okay, so it is stopping here. It is stopping because ATP is needed. ATP is needed here to convert it, right? But already the ATP is utilized. ADP, I mean, here it needs only AMP. One AMP it has to remove and ADP should be added. So once the ADP is added, it's converted to the PRPP, right? But here, but here, when there is a sufficient number of IMPs or GMPs are present, then it won't be added here. So when the ADP is not added, then PRPP will not be synthesized, right? And even in this process also, PRPP is converted to 5-phosphoribosylamine, right? Here also, when there is an excess amount of the purines, then AMP will not be added in these steps, GMP will not be added, IMP also will not be added in these 9 steps. Okay, then IMP will be converted to the XMP and GMP, right? So when there is a more GMP, then the GMP will be acts as a feedback mechanism so that the IMP will not be converted or it won't create the XMP here. But means there is a feedback mechanism is there. Regulation of the purine nucleotide. Clear? Now, there is one more method called the salvage pathway. So what is the salvage pathway? I have shown you with a diagram, right? So this pathway ensures the recycling of purines found by degradation of the nucleotides. It's a recycling. We are not creating new purines here. Okay, you are utilizing the breakdown nucleotides from the nucleic acids. Okay, nucleic acids. So nucleosides and deoxynucleosides can also be salvaged. Nucleosides also. What is a nucleoside? Means the phosphate will be separated, then the sugar will be separated, and the purine pyrimidines also will be separated. The purines can be directly converted to the corresponding nucleotides, and this process is known as salvage pathway. Okay, we are not building, right? So just we are utilizing the. I mean, there's nothing but the recycling. Okay, then this is a salvage pathway. So what is the salvage pathway here? We have already ready-made adenine is there because we are utilizing the recycled one, right? So adenine is there. So using an enzyme adenine phosphoribosyltransferase, transferase, then it is linked to the ribose sugar, so it's converted to the AMP. Then guanine, the same hypoxanthine guanine phosphoribosyltransferase, transferase, it's linked to the a ribose 5 phosphate. I mean, it's we are linked to the sugar with phosphate, and it's converted to the GMP. When there's a hypoxanthine also IMP, right? IMP, hypoxanthine. So when these are available, then we are converting them into the IMPs. Hypoxanthine guanine phosphoribosyl transferase. Right? This is a salvage pathway. In the salvage pathway, no need to create the I mean, the atom-wise molecules, right? Or purines. We are not going to create any purines part by part, right? Now, coming to the catabolism. Catabolism is the breakdown. The breakdown means initially we learned how to construct. Now we should know how to break down. So what's the breakdown? Adenosine will be broken down into inosine. It's a reverse, right? It's a reverse inosine. Then inosine to hypoxanthine. So here guanosine is converted to the guanine. Then guanine, xanthine. Then here hypoxanthine also converted to the xanthine. And finally, the xanthine is converted to the uric acid. So that's the reason uric acid is a byproduct of, I mean, uric acid is a product, end product of the purine metabolism. Okay, if you take the purine, okay, then adenosine will be converted to the inosine, inosine to hypoxanthine, then uh, hypoxanthine to xanthine, then guanosine to guanine, then guanine to xanthine. Okay, finally, it's converted to the uric acid. Okay, it's a catabolism. So I mean, it's a catabolism, but if you want, write all the steps in detail, then you can use this one finally. Okay, the molecular structure you can write GMP, guanosine, then guanine, finally xanthine to uric acid. Then here adenosine, inosine, then hypoxanthine, then xanthine, finally uric acid. The same thing. Okay, the same thing, but here I have written, but here we are using the molecular structures. Then coming to the disorders of the purine metabolism. So what is the disorder of the purine metabolism? See, what is the purine metabolism here? Purines, when the purines are metabolized, then the uric acid will form. Okay. So here the metabolism means maybe the construction or maybe the destruction. So uric acid is the end product of the purine metabolism in human beings. Okay. So the normal concentration of uric acid in the serum usually in the blood is 3 to 7 milligrams per dl. 3 to 7 milligrams. So usually in women it will be slightly lower. Okay, 1 milligrams like that. The daily excretion of uric acid about 500 to 700 milligrams. Daily excretion via urine. Okay, because in the urine contains uric acid also. Some animals, they excrete completely uric acid. We excrete urea, right? We excrete mostly urea, but uric acid also we excrete, that is 500 to 700 milligrams. But some like lizards, some insects, they release more uric acid. Okay, fine. Hyperuricemia. What do you mean by hyperuricemia? Excess of excretion of the uric acid, or excess formation of the, or excess present of the uric acid in the blood. That's called hyperuricemia. Hyperuricemia refers to an elevation in the serum uric acid concentration, usually that is 3 to 7. But if it exceeds 7 milligram, this is sometimes associated with increased uric acid excretion also, uricasuria. So what is the metabolic disease? Gout is a metabolic disease associated with the overproduction of the uric acid. So at the physiological pH, uric acid is found in a more soluble form of sodium urate. Actually, in the uric acid is present in the See, you know the extracellular fluid, right? We already discussed about the extracellular fluid. Extracellular fluid contains more sodium, right? So at a particular pH, so whatever the uric acid is found, that is soluble and will be linked to the sodium. Okay, it's combined with the sodium, uric acid, and it appears as a sodium urate. Sodium urate is nothing but the sodium uric acid. Okay, that is a soluble form. Okay, when it is in soluble form, then what happens? It will get easily excreted, right? If it is unsoluble form, then that is a problem. See, in severe hyperuricemia, crystals of sodium urate or sodium uric acid, okay, gets deposited in the soft tissues, particularly in the joints. Okay, this is a soluble form, right? But when the uric acid levels are more, then it is combined with a sodium, which is in the extracellular fluid, right? So that is deposited in the soft tissues, particularly in the joints. So such deposits are commonly known as tophi. Tophi. This causes inflammation in the joints, resulting in a painful gouty arthritis. So sodium urate or uric acid may also precipitate in kidneys and ureters that results in the renal damage and stone formation. Okay, like this. So this sodium urate may form, okay, here in the place of joints, causing gouty arthritis. This is called the gouty arthritis, pain. Okay, uric acid crystals along with the sodium. See, uh, one example I will show you. Yeah. So first, what is happening? Uh, see, yeah, crystals formed, uric acid crystals formed. So where in the in the joints, okay, mainly in the joints and the legs, okay, crystals form. What type of crystal these are? Sodium urate. Sodium urate crystals will form. Okay. Then white blood cells attack because this what we say, it's not normal, right? It's abnormal thing only. So white blood cells attack. Then crystals pop the cell. What the crystals do? It will pop the cells. Then cell release proteins. What the cells do? It will release the proteins. Cytokines. Lot of proteins will get released, right? So cell releases proteins. Then these proteins call in more white blood cells and cause inflammation, right? Inflammation. Then proteins lower pH, making it possible for more crystals to form again. Okay, so this is the cycle. So once the inflammation happened, then the swollen, the legs may swell, right? Or the joints may swell or swell, right? Okay, let us see some more content about this gout. Historically, gout was found to be often associated with the high living, overeating, and alcohol consumption also. I mean, why more uric acid crystals will get deposited in the, you know, uh, joints means overeating. What does it mean overeating? Overeating of non-vegetarian. Non-vegetarian means salvage pathway. Sometimes, the nucleotides will broken down and finally those purines will be liberated. Then the end product of the purine is nothing but the uric acid only, right? So if you eat more non-veg, then the purine metabolism will happen. So that is one indication of having high uricemia, right? So high uric acid in the blood. Then actually, in the previous centuries, alcohol was contaminated with lead during its manufacturing storage. Usually, in the creation of the alcohol or in the preparation of the alcohol, in the olden days, they used lead. Okay, so lead poisoning leads to kidney damage and decreased uric acid excretion, causing gout. Even nowadays, also, the normal non-standard alcohol drinks contains the lead. So, what the lead do? Lead poison will cause the decreased excretion of the uric acid. So that the uric acid, if it is not excreted through kidney, then that will be deposited in the joints, causing the gout. So that's the reason eating high meat and consuming the alcohol also will leads to the gout. Okay. So gout is of two types. One is a primary gout, the other one is secondary gout. So what is the primary? Primary means we cannot do anything. There may be inborn error. That's inborn error. Metabolism. Okay, there's a error in the metabolism. That's a byborn only. Okay. So why? There are some enzyme defects. PRPP synthetase. What is the PRPP synthetase? That is a phosphoribosyl pyrophosphate that we use, right, when in the synthesis of the uric acid, right? I mean, in the synthesis of the pyrosin, I mean, the purine. So we use this enzyme, right? When there is a defect of this enzyme, then the purine may not be synthesized, right? Purine may not synthesized. So PRPP, glutamyl amido transferase, then HGPRT deficiency. So the lack of I mean, these enzymes may be due to some genetic problems or maybe inborn errors. Okay. So the defect of these enzymes may cause the primary gout. So it's the primary. What's the primary? Primary means inborn. Okay. So what is the secondary? Secondary means after getting maybe disease or after consuming more alcohol, after eating, high protein, I mean the nucleotide content. So that is secondary gout. Okay. Secondary hyperuricemia is due to various disease caused, maybe due to cancer also. Right. So these are the secondary things. Okay. Increased degradation of nucleic acids. Okay. More uric acid formation is observed in various cancers like leukemia, polycythemia, lymphomas, psoriasis. Okay. Then sometimes, renal impairment also. When there's a renal impairment, then it fails to, excrete more uric acid. So that is also secondary gout. So we have two types of the gout, primary and secondary. And coming to one important thing in the primary, so there is a enzyme deficiency. That enzyme is called the hypoxanthine guanine phosphoribosyl transferase, HGPRT. What is this called? HGPRT. So in some people, HGPRT deficiency will be there. So that comes under the primary gout only because it's an inborn error. Inborn error. Okay. So this is an enzyme of purine salvage pathway and its defect causes Lesch-Nyhan syndrome. What is Lesch-Nyhan syndrome? This is one syndrome. Actually, Les is a student, medical student, and Nyhan is uh, like maybe the research guru, okay, or maybe his professor, right? So both the student and professor they invented this one. They observed this one. Okay, so that is the reason on their name this syndrome came, Lesch-Nyhan syndrome. Okay, so this is an enzyme of purine salvage pathway. What is an enzyme? HGPRT. This disorder is associated with increased instance of purine nucleates by a twofold mechanism. First, initially, what happens? Hypoxanthine and guanine say decrease utilization of the purines by salvage pathway, resulting in the accumulation of diversion of PRPP for purine nucleotides. Usually, the recycling should happen, right? Okay. If there is no recycling is happening, then what happens? Whatever the hypoxanthine and guanine formed after recycling, these are not utilizing by the salvage pathway, right? So the accumulation of the hypoxanthine and guanine is happening. Okay, so when the hypoxanthine and guanine the accumulation is happening, then automatically the uric acid levels will increase. Okay, then secondly, the defect in salvage pathway leads to decreased levels of IMP and CMP, causing impairment in the tightly controlled feedback regulation. So sometimes what is happening here? Even the salvage pathway also, IMP and CMP, okay, decreased levels. So these two are the decreased levels. Okay, so that is the reason what is happening here. There is a problem in the feedback regulation. Okay, when there is a problem in the feedback regulation, what happens? So automatically it will increase, right? It will increase the production. Okay, so that is the reason in the Lesch-Nyhan syndrome, Lesch-Nyhan syndrome, the accumulation of the uric acid will be more. Okay, one second, I will just explain this with a good example. Yeah. What is this Lesch-Nyhan syndrome? So this is one short question they may ask you in the examination. This disorder is due to deficiency of hypoxanthine guanine. Okay, phosphoribosyl transferase. That is called the HGPRT, an enzyme of purine salvage pathway. This is a sex-linked metabolic disorder. Usually it happens only in the male, okay, as this is a dominant one. It affects only the male and is carried by excessive uric acid production. What happens in this syndrome? Mental retardation, aggressive behavior, learning disability. Okay, so the patient of the disorder have an irresistible urge to bite their fingers and lips, often causing self-mutilation. Yeah, I think they may ask one case study also. Okay, they may ask the case studies also on this. I think 1 second. I had gone through one case study. Just a moment. Yeah. See, a mother of a 4-year-old boy, 4-year-old boy, is worried about his failure to develop, his aggressive behavior, complaints of pain in his joints, and his compulsive urge to bite his fingers and lips. During history taking, the mother revealed about the boy's maternal uncle having similar history. The case was referred to a nearby medical college hospital for further investigation. Okay, so how would you investigate? See, his maternal uncle is having means that is hereditary. He is having some inborn problem, and pain in his joints means that indicating that the accumulation of the uric acid, and his compulsive urge to bite his fingers and lips. Okay, so that's aggressive behavior as well as I mean there is an irresistible urge, okay, to bite their fingers and lips. Okay, sometimes they may cause their self damage, right, self mutilation. Okay, an estimation of serum uric acid level should be needed in this case, as this revealed a concentration of 10 milligrams. Actually, 7 is the maximum, but that boy is having 10 milligrams, showing that hyperuricemia. The clinical picture, along with the elevated urate levels and a suggestive family history, clearly point to the presence of Lesch-Nyhan syndrome, an X-linked recessive disorder with abnormal neuropsychiatric symptom. It's an X-linked recessive disorder. Okay. Yeah. So there is one, clinical case-based learning also there about this Lesch-Nyhan syndrome. Okay. And yeah, then some more explanation about this gout. See, coming to the gout, we already discussed. There are two, primary and secondary. Secondary means it is due to various diseases, right? Various diseases are taking, based on the diet, accumulation of the uric acid, based on diet or based on alcohol. Okay, or maybe we get different diseases, right? So that is secondary one. Primary one is inborn error. Okay, already I told you in the inborn error means there are some enzyme deficiency, PRPP synthetase, PRPP glutamyl, okay, HGPRT deficiency, glucose 6 phosphate deficiency, elevation of glutathione reductase. Okay, see here, I made a diagram, but I think this is not that much required. It's more than enough for you. So glucose 6 phosphate. Okay, the problem is here with this enzyme. Then glutathione reductase here also. If there is any problem in the increase of this enzyme, here PRPP synthetase, this enzyme, this enzyme, HGPRT, these enzymes also will lead to the primary gouts. Okay, when there is any problem in these enzyme secretions, then the primary gout will come. Okay, then this is associated with defects in purine metabolism. Already explained this one. Hyperuricemia, gout also we discussed. Lesch-Nyhan syndrome also we discussed, and kidney stones. Even kidney stones also. Why? Because the accumulation of the uric acid crystals, maybe in the ureters or in the bladder also. Hyperuricemia means just now we discussed 3 to 7. If it is exceeding 7, then that indicates hyperuricemia. In the female, that should be 1 milligram less. Okay. Then primary hyperuricemia means an innate deficiency. This is nothing but the primary. Primary means inborn. Secondary means after born, after birth, right? maybe due to different factors. Then progression of the hyperuricemia to gout. So how this hyperuricemia convert to the gout? Already we discussed this one, right? Initially, asymptomatic hyperuricemia. Initially, there, I mean, the patient he won't see any type of the symptoms, asymptomatic, right? If it is greater than 6.5, okay, urate crystals may start to deposit in the joints. No evidence that treatment is required. Okay, means if it is more than 6.8 or 7 milligrams also, but slowly the deposition may happen in the joints. That time, no evidence that treatment is required. Then acute gout. Insufficient uric acid deposits, I mean, if sufficient uric acid deposits develop around the joints, and if the local environment or some trauma triggers, maybe, the inflammation, okay, the release of crystals into the joint space and inflammatory response occurs. These flares can be self-resolving but are likely to recur. Again, they may recur. Then intercritical periods. It's an acute one. Then these are the intervals between attacks. During these periods, crystals may still be present at a low level in the synovial tissue and fluid, resulting in the future attacks also. And no need, okay, no need of this one. Advanced gout means how this hyperuricemia is converted to the gout. Okay, I mean, I think it's no need because it's not that much needed. Then gout causes under excretion of uric acid leads to accumulation of uric acid in the blood. Then diet rich in purine alcohols, deficiency in dietary products, I mean dairy products. Then increased purine degradation, increased PRPP synthesis activity. Okay, then decreased or partial HGPRT activity. So these all leads to the gout only. Then what is the treatment? Then I mean colchicine, reduces the inflammation. Then allopurinol, it's a very good. Okay, it will inhibit the uric acid in there. So whoever suffering from the, this uric acid gouts, then they were usually treated with the allopurinol. It's a famous medicine, and everyone knows this one. Okay, allopurinol. Then low purine diet also they suggest foods that are high in purine include red meat and organ meats, yeast and yeast extract like beer and alcoholic beverages. Then asparagus, spinach, beans, peas, lentils, okay, cauliflower and mushrooms. Avoid caffeine and alcohol. Keep hydrated. Why hydrated? Because we had to excrete more uric acid. So take more water to eliminate that uric acid, okay, through kidney, not in the form of excretion. So treatment for gout. Then these are the kidneys. I mean kidney stones, right? Uric acid crystals. Okay, they may precipitate here, maybe in the uh, ureters, or maybe they are in the kidney, or sometimes they may obstruct the bladder also. Okay. Then, yeah, I told you about this allopurinol, right? A suicide inhibitor used to treat gout. Okay. See, it's hypoxanthine, right? Hypoxanthine. So hypoxanthine will be converted to AMP. AMP, right? So to stop the conversion here, the allopurinol is inhibiting this one. Okay. That is the reason it won't be converted to AMP or other. See, xanthin oxide, xanthin urate. Usually this xanthan will be converted to finally uric acid. See, when you see the degradation, this hypoxanthine to xanthin, then xanthin to uric acid will form, right? So to stop the formation of uric acid, here the allopurinol is inhibiting, okay, the action of this enzyme to convert the xanthin to the uric acid, which is the end product. And one more thing that's called a severe combined immunodeficiency also there. Severe combined immunodeficiency. Severe combined immunodeficiency means simple here. Adenosine deaminase deficiency. This is a enzyme deficiency. Okay. See, we need the purine. Why purine? Why purine and pyrimidine? To create the nucleotides. So when you create the nucleotide, then the cells will get divided. When the cells will get divided, then the muscle will get, divided, and the human will grow, right? So we need the nucleotide. That is very important, right? But there is a problem here. In some children, it's maybe it's a hereditary severe combined immunodeficiency syndrome. Severe combined immunodeficiency means adenosine deaminase deficiency will be there. Accumulation of DATP inhibition of ribonucleotide reductase equal to B and T is unable to divide. So because of this adenosine deaminase deficiency, what happens? The child, he cannot proliferate, or he cannot manufacture the B cells and T cells. Or the B cells and T cells will not be produced in his body. What happens if B cells and T cells will not produce? B cells and T cells are useful for the immunity purpose, right? Immunity purpose. Okay. So if the person is having no B cells and T cells, then how he will survive? He cannot survive, right? Then there won't be any inflammation. Then he cannot survive. So these type of children may die within a year or below a year, right? So there is one case also there that's called the bubble baby disease. We also call this the bubble baby disease. Can you see this bubble? This plastic bubble is there. So actually, I will read this one. Children affected with SCID, means severe combined immunodeficiency, appear perfectly healthy at birth, but they are extremely susceptible to infection. Why? Because in their body, B and T cells were unable to divide, right? So most babies born with this disease die before the age of 1 only because of some infections. The standard treatment of SCID is a bone marrow transplantation. That's it. Because in the bone marrow only the B and T cells will proliferate, right? So that is the reason we have to do only bone marrow transplantation. That is the only permanent solution. Okay. So the name bubble baby disease owes its origin to a 1960 real life incident of a boy named David Vetter. Actually, a mother, a desperate mother, she had a child. Her child name is David Vetter. It happened in 1960. Okay. So she protected her kid in a plastic bubble for so many years. Why? Because not to get affected with diseases, right? If any disease, attacks, he cannot survive because he's not having the B and T cells, right? He is completely lack of immune system, right? So we cannot protect him, right? But that lady desperately he protected his kid. She protected her kid till the age of 12 by keeping in a bubble. Got it? So there is a separate movie also there, a boy in the plastic bubble. The boy in the plastic bubble. Okay, I think that movie released in 1976. The boy in the plastic bubble. It's based on the David's touching story. The boy name is David. Okay, so this adenosine deaminase deficiency causes this severe combined immunodeficiency. Okay, so that is also the primary one. Then how to treat? Already I told you allopurinol. Okay, then when there is any pain, then we should use the colchicine. Then drug therapy also we need. Anyway, some corticosteroids to prevent the, what we say, temporarily, and the steroids also we have to give. Okay, so that is about the purine metabolism. Okay, fine. So time up. So what we do tomorrow, we'll be discussing the pyramidal metabolism. Anyway, this is a very short one only. Okay, so just I think 15-20 minutes is enough to finish this pyramidal metabolism, and I'll be sharing you this PPT and the today's recorded session also. Okay, and I think I missed the immunology also because that day